lab, and I
Do you not want to be
5 to 10 minutes putting on gloves because they were like still wet from washing my hands. So like the whole time they were in there, I was trying to put on gloves.
Well,
like,
the
it
company's
wasn't
running
easy.
the bill. You're not graduating from Northwest.
Oh,
That's
okay,
totally company.
I just
That's nice.
they paid for it. No, but like it wasn't that wet. Or like I tried them in the past.
There's-
They were soaking
there's-
wet, but
you don't
and
get
so
this from
it just looked like I was like
Northwest.
standing there doing nothing.
Probably the
Embarrassing.
depends on how often you
There
actually
wasn't
use
enough.
the work.
And I wasn't going to stand there, but I'm like, oh, just go back in. And then since we put on, I wasn't even going to put on my gloves, but she was there, so I felt comfortable. Because I could just use like this. So I was like, for her box. So that was like 10 minutes.
Are you having trouble with that?
The output is, so this is how you use
I don't
it.
have it.
This is last one.
Right.
This is a
I
gift.
don't know. I didn't even know she was teaching.
You just sit.
Corey? Are you kidding?
So I try to play around with things. So,
Pretty good, how are you doing?
Okay. While the computer's loading here, so this should be familiar to all of you. Got translation. We got transcription. We got processing. We got translation. We got modification. We got degradation. Proteolysis, we'll call it. What would this be? Nucleolysis? DNAlysis? Packaging? Well, Hector's funny joke. Ready? The National Dyslexia Association. Okay. Talking about gene regulation. And this is the central dogma of biology as you should be able to draw it when you walk out of this class. Okay? Came in, it was 3. It was DNA, RNA, protein, and you had 2 words over the lines. So this is more complicated. Hopefully you've got all of this as standard thinking now. I'm going to talk to you about some things that happen in this process which were discovered. One of these is microRNA. I'll talk a little bit about the machines in the cell that do this. Before we get to that though today, my word, it's taking a long time to load Windows. Before we get to that, I want to talk about I want to show you some data. You're not allowed to answer. And because we've talked about it, I'm hoping that I can get some of you to figure this out. So, my gene that I was studying when I was in graduate school, I discovered this gene. I found, so let me draw this out as you guys would do in this laboratory. I ran a PCR reaction, and I had back plus everything. Nucleotides, primers, all this stuff. What I got was this stuff here, and this band here. On the DNA molecular weight marker, not ladder, marker. The size marker. This is 50 nucleotides, maybe 25 nucleotides, short. This is 600, okay? You think you know what's going on here? Right. We did, what did I call it? We call it CNF. Somebody somewhere else has called it TMEM 106C. Because the gene. Huh? It's yours? Oh, I discovered that. Right? Someone else discovered it independently. I discovered it first. This is no tag. So. I'm just going to say plus means everything is in there. And what I got in that case was I expect my gene to be about 700 nucleotide, 700 base pairs size. Someone in word can just put into words what you visually see on this gel. What do you see? Everything worked out as it should.
Got primers, got things where you expect.
Okay. So my positive control is back. I know back is going to work. I got the expected size. I had primers. A little left over. That's fine. In my negative control, this should not have worked. I had primers, nothing. Finally. And then in my actual condition that I care about, I had a product and I had what looks like primers. Does anything bother you about the product? Two bands.
It's a little weird band. I guess that would be something to look at.
Okay. What does it mean? You guys want to get in groups and talk about it? Sounds like fun. All right, I'll give you guys 2 minutes while I get my file loaded up here. Right? I'm watching. 2 minutes.
5 minutes. That's just another thing, What do you look at, Ricky? That's something that was
Who needs another minute? Who needs 30 more seconds?
No.
No? You're there?
I mean, we have something that felt kind of funny.
All right, sounds funny. I'm all for jokes. I love it. Make me laugh.
It's not really a joke, but.
You want to go first then?
Sure, maybe something about like the primers. Like you put primers on and maybe they just like keep adding on to a few of them and then you get like a longer strand because your primers are adding a little bit of length.
So one primer hybridizes to another primer, and then the tack extends that. And the tack extends that one. This is what you're saying?
Yeah, something weird like that.
So that when we separate this, I have this. And then that can hybridize like this, right? So we can add onto that, we can add onto that.
This is like only happen like very few times. But like since you replicate it, something like that.
Okay, that's great. How would you control for this?
Change your primers. I don't know. Wow.
There's a way up.
Increase your temperature so that it doesn't anneal on that.
What goes into a Someone want to come up and write the formula for a PCR reaction up here? Yeah. Formula? Huh? The recipe. You open up the cookbook and there's a recipe. And 3 cups of TAC.
It's like less than
You may use your notes. All right, let's blurt it out one at a time. We're going to start over on the side that's quiet. I won't pick. I'll from this side over. Blurt it out. I want to hear it.
Template,
Template.
right primer, left primer,
Primer 1. Primer 2.
buffer,
Buffer.
DNTPs,
VNTPs.
TAC,
TAC.
water.
Okay. If it's primers that are binding to each other and amplifying, what do I need in a PCR reaction for that to happen? What can I remove from the standard reaction and this would still happen? Template. If I run this PCR reaction, if your hypothesis is true, what are my results?
The same as plus CNF.
It'd be the same as this. So, this is great. So, we would get primer down here, and we'd get, let's say I get that. And this is the mystery band, and what's this?
Good stuff.
Huh?
Good stuff.
That's the stuff I want. That's my PCR product. Okay, great. So, I'm hoping that you guys are seeing. This is stuff I actually faced. I walked in and I was like, what the hell is this? So first, when I ran it, I just had a really big smeary band. I was like, oh man. So I went out and I got a gel and I poured an agarose gel that was like that big. And I loaded the sample and I ran it like in 6 hours. So my bands ran all the way down. It was like 3% gel. So it just ran forever to get through this gel, and it actually separated out into 2 bands. One really, really intense like this, and then a really faint one like that. That was great. So I had no idea what it was, and that's one possibility. I think in reality what would have happened is, what I showed this guy hybridizing like this, that thing might actually have hybridized like this. And so it would have amplified a little smaller than this guy, right? And that's called, what you described is a well-known phenomenon called primer dimers. And primer dimers were something that the computer program, when you were designing your primers at the start of the year, the program that you were asking to design your primers said, I want to make sure I don't have primer dimers. And so it excluded all of those. You didn't know it at the time, but you just learned it. If primer dimers had happened, what you'd actually see is something like this. Does that make sense? This is 2 primers in size. This is 3 primers in size. This is 4 primers in size. This is 5 primers in size. Just work good. Okay? Awesome. That was really great. I considered that possible outcome. Who came up with an alternate explanation for this? You guys want more time? Yeah.
Could you have accidentally added some of your back primer in?
Okay, that's a great one. Yeah, because it's the correct same size as back. An error on my part. I pipetted, and then I did back. Then I didn't change my pipette tip. So then when I got primer for TMEM 106C, just a small amount of the back primer was still on the inside of the pipette tip. And I transferred that in. And I did that mistake a couple times, but I wound up that this is back, and this is TMEM 106C. Great. So I could order new primers, and I could repeat the experiment, making absolutely sure I don't do that. And when I do that, the experiment comes out. So somebody got results like this and figured out what it was. This is research. This is what I call, pardon me, this is what I have been saying to you, you're at the end of knowledge. A couple times in the class you've asked me a question and said, I don't know. You're at the end of knowledge. At one point, there was a person who got that as a result. They went to their advisor and said, what is it? And their advisor said, I have no idea. And you're supposed to be getting your PhD, so you need to figure that out on your own. Let me know when you got it. So that day when we went into lab and I said, here are the results, you guys figure out what you're going to do, that's it. That's research. It's a pretty scary thing, isn't it? But it's also pretty cool. If someone's going to figure out what that is, it's going to be you. Or me. Okay. So I'm going to talk about, I'm going to lead you on here into one possible explanation. TMEM 106C. Does anyone have something very similar to that?
TMEM 106A.
You have TMEM 106A? A? TMEM 106B. So it turns out, in the human genome, there are 3 genes that have very similar DNA sequences. With that, could I just lay it out for someone what we're looking at here? Maybe the primers designed for TMEM 106C also were able to PCR amplify EM 106A or B, right? And the gene isn't the same size, yeah? It would make sense then that the primers I designed for C would work really well, and the primers I designed for C wouldn't work very well on A. You'd get a little bit, but not much, or B, or whatever. So this is definitely happens. People run a PCR reaction, and the primers work on more than one gene, and you PCR amplify 2 genes out of it. Okay. No one's got the other possible solution? Alex, you wanna show how smart you are?
What are the possible solutions?
Another possible solution. You know?
I sort of had the first one we came up with, parts of it, but
Yeah.
I don't know. As far as I got, there's another way.
It's the actual explanation. And Alex got it, right? You got it on your gel.
Yep.
He got that on his gel.
Oh boy.
Could it be that the Taq polymerase is starting to degrade?
Sure. Could it be that the PCR product is full and then the Taq polymerase is sitting there kind of bored and it's like, I think I'll just take a few bites out of this here. If that happens, generally what happens is it nibbles like 10 off of this one and then 200 off of this one, and you get a smear DNA. All right, what was the explanation I sat down and shared with you? Do you remember?
I remember what we did about it. I lowered my TM,
Okay.
and so that it would have more time to hybridize.
Okay. So be more specific.
Yes.
And what was the reason? Do you remember the phenomenon I thought might be happening?
I recall discussing it, I just don't remember for sure what
Great.
it was. So.
This will be an amazing opportunity to refresh your memory.
Yes.
The primer fell off?
The primer fell off? Yeah, possibly. So, let's get into this. Sorry to put you on the spot there.
That's all right.
I can't erase that. Let's erase this over here.
Now,
Okay, you missed a chance, someone else. Someone know what I've just drawn up here?
Cut sites for an intron.
The what?
The cut sites for an intron.
The cut sites for an intron. So what's this over here?
X1.
This is exon 1. What's this?
Intron.
Intron 1. What's this?
X1.
Exon 2. So do I throw away that or do I keep that? Keep it? Keep or throw away?
Throw away.
Okay, great. So what I'm going to get, let me do it this way. So what I'm going to get is this, connected to this, right? Where I throw this stuff away, so that I'm going to get C, A, G, G, right? That's this G right here, and this is the C, A, G, right? When we talked about splicing before, I said there's the spliceosome, or the proteins that do this. And the way they find the spot to cut is they have what they call a guide RNA. It's a really short RNA called a SNRP, SNRP, remember that? And they hold that, and that thing hybridizes to the sequence, so it's complementary to this sequence. Does anyone remember when you were characterizing the splice sites in your sequence, did anyone have a perfect consensus sequence in there?
I think I did, actually.
You think you did? Yeah, probably some of you did at some of your spots. Did anyone have a sequence that was like super crappy and you were amazed? Yeah. Yeah. So the way we characterized, the way we drew this out is we said, okay, the consensus sequence is CAG cut GTRAGT. So if my sequence was perfectly that, the way we characterized it is we put a plus for each one of these, remember? Remember doing that? We did that with the Kozak, we
It
did
was
that.
like 8 out of 10 or something pluses.
Yeah.
Real close.
What happens if the sequence there is not the consensus sequence? What if my sequence is this? Okay, that's good. That's good. Whoops. There we go. That's good. That's good. That's good. That's not a very good splice site, is it?
You have to change the 2 G's.
Yeah. Okay. Well, let's be kind. Let's say it's a G.
Almost.
Feel better now?
A little less.
Okay.
Might work.
What if right here I have a sequence which is this? All right? So the original one was that, and this one is now plus, 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 no, 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 plus, plus. Well, this one's, I'm going to say, we're going to be half empty today. This one's equally as crappy as this one. Right? And this sequence is located right here. So when the splicing machinery comes in with its guide RNA to find a sequence to hybridize to, it goes here and it says, I don't really hybridize very well there. And it looks around and is like, I don't see anything better. So we're going to hybridize here, we're going to set up shop here, and we're going to cut at this spot here. And then on a different RNA on the other side of the cell, splicing machinery comes in and goes, this looks pretty good, let's cut right here. So RNA 1 gets cut like this, and it's missing this stuff. Does anyone know the word for this? Maybe you've learned this already in some other class. Alternative splicing. If I were to run a PCR reaction on this, they come out the same size. Nope. One would be shorter than the other one. Does that make sense? Holy shit. You guys awake now? And do you want to hold this? Just kidding. I'm going to sit down for a second. Okay. So alternative splicing is for real. CAMEM 106C, maybe B or A, I don't remember which one it is definitely alternatively spliced. When I was in graduate school, I cloned both versions of the genome. I want you to think about this. One sequence in the DNA genome produces two different messenger RNAs. And let me tell you about this particular gene. You're going to love this. I'm probably going to regret doing that. So if the code for one is this, and the code for the other is this, such that this stuff is present in one and not the other. Oops, ATG. When the ribosome binds to the cap and scans down to the ATG and begins to translate, this protein will be longer than the other protein. Right? This guy has amino acid coding stuff. And it turns out in this particular gene, CNF, TMEM 106C I think it was, this is a multiple of 3. What does that tell you about this? Why is that important?
It's going to be the same amino acids after that.
It's in frame. So basically what this guy has is 12 extra amino acids in it. But this sequence here is the same as this sequence here. And this sequence here is the same as this sequence here. This protein is just a little bit larger. When I took this amino acid sequence and put it into a computer program, and the computer program said, huh, what you've got is right in the middle of your protein, you have a whole bunch of hydrophobic amino acids that do not want to be in water. Where do you think they want to be?
Cell membrane.
In a lipid membrane. This guy doesn't have those amino acids. So what the computer spit out to me is said, I predict that this thing is in the mitochondrial membrane. But when I put this sequence into the computer program said, I think this thing is hanging out in the cytoplasm. Two totally different proteins from one sequence of the DNA genome. So I made antibodies, I made dyes that would stain these. And when I took that, when I cloned this, and put it into cells, and stained cells, the mitochondria were green. When I took this guy and put it into cells, the cells all over the place were a little bit hazy green, but they were not distinctive dots. Nobody knows what it does. I think it's a cancer gene. Who thinks this is really cool stuff? All right, so this is just kind of my way of, I did a chalk talk here because I think it just had more effect, and because I'm trying to move you guys into a situation where you can look at data and make your own conclusions from the data. This is an upper level biology course. Let's start doing these courses. But the first section of this course, or the first section of this PowerPoint file, walks you through alternative splicing in basically the same terms here. There is this thing called the spliceosome, and it uses this SNRK to find the site. If the site that it hybridizes to is imperfect, it may look for an alternate site. Sometimes it's this spot up here, where it's locates at a different spot. Sometimes it's the machine that sits downstream that finds a different spot. I think usually it's the downstream one that finds a different spot. Okay. This just illustrates, here we have our cap, we have our messenger, our RNA. This is the pre-RNA, it's been capped, it's got a poly A tail, and now it's about to be spliced. And in this case, the upstream site could be either here or here. Turns out if it splices this way, there's a termination codon in here. If it splices this other way, that termination codon gets spliced out, such that this is protein code, right? In this version, this stuff is attached down there, but this is not going to be translated, is it? Because termination will have been hit. You see how the two termination codons? There's a termination codon here. I didn't put it in because it's not used. In this twice version, the termination codon is hit early. There's a different termination codon hit early. It's not here because it's been spliced out. Right? So there is a very important gene in the human genome that this happens. And in that one, this is a transmembrane domain that sticks the protein in the membrane. And when that gene now suddenly starts getting alternatively spliced, it's no longer put into the membrane and it's floating around. And that gene is the B cell receptor. Any of you in immunology? What's another word for a soluble B cell receptor? Antibody? It's part of your immune system, critical part of your immune response. Okay. You guys want to talk about this? Anyone have questions about this?
So do we know if this is regulated by the cell at all, or are we just kind of assuming that it's random?
That's a great question. We haven't yet got to that. You're three slides ahead of us. So let's answer that. The question is this just kind of random? Like, hey, I found this splice site, so that's where we're going to splice this time. Or does the cell control it? The cell controls it. So the cell, actually, if the cell had, I should not have erased it. The cell may have a gene, and the whole purpose of the gene is to bind to this sequence right here, such that if it binds to that sequence, the spliceosome with its guide RNA can't possibly find that sequence to splice to. And so its only option is to splice to here. And that's what happens when a B cell gets activated and becomes part of the immune response. It makes a protein, and that protein binds to this. So now the only version you can make of the B cell receptor is the soluble form that is called an antibody instead of a soluble B cell receptor. So that would be something that sits on there as a splicing silencer. You could have something that sits next to it and helps the splicing machinery come in and identify that, in which case we'd call it a splicing enhancer. Great question. Yeah. In some genes, in some cells, it may be random. Some of this and some of that will have a little of both. But in other cells, it's very specifically controlled. Estimates are that 70% of all human pre-RNAs are alternatively spliced. In liver cells, it may be form A, and in neurons, it may be a different splice form that happens. 70% of the genes. So if there's 25,000 genes in the human genome, in the DNA, that means that there's 50,000 different proteins that can be made from that. Some genes can be alternatively spliced a dozen different ways. From one DNA sequence, you can get 12 different proteins.
Is there a way or are there mechanisms that you can splice out like that amino acid sequence after it's been coded to make it the other form of the protein?
So the question is, can we, I'm going to try this, can we get the mature RNA and make the protein, then have an amino acids, the 12 amino acids cut out and stitch that back together? To the best of my knowledge, no. And I suspect if that existed, someone would have found it. There would be evidence. Someone would have found something weird on a gel. Other comments or questions on this? Okay. Splicing enhances splicing sound. Okay. Let me tell you the master of this. It's a good setup for the next. Anyone ever hear of this virus called HIV? Human immunodeficiency virus. So we can write the genome of HIV out. We can just illustrate it like this. There's a sequence in HIV. I'm going to get the details wrong, but don't hold me to it here on the camera. I have a friend in grad school who studied this stuff. He'd kill me because I forgot his research. So, +1, so we get this transcript off of here, like that. And this thing can be spliced. Like this, right? Put this together. That. We have an ATG, we have a termination codon. It can be spliced a little bit different. It could be spliced there instead. Such that we get not a multiple of 3. What does that mean? It shifts the reading frame. So it does, this is definitely the same as this, right? Or the first part here, right? Yeah. And then we're out of frame. So this is different than this. Do we go back in frame in this tail part? Nope, it's still out of frame. So a different termination codon is used. The amino acid sequence, so in the original example I was talking about is, the protein is basically all the same amino acids, you just 12 are not in there, because they got spliced out. In this one, the first 7 amino acids are the same, and then the protein is totally different for all the rest of the amino acids. It's not that you've got, one protein's in the mitochondria, and then it's basically the same thing, but it's in the cytoplasm. No, these are 2 totally different proteins coded in just one part of the genome. This is called NEF, and this is called TAT. One is involved in packaging and escape from the cell, and one is a transcription factor. And it turns out this guy can actually be alternatively spliced get two different versions of TAC.
So it's 3 different from 1?
It gets better.
No, we can go backwards.
So another strand, you make this RNA. And that codes for a protein if you're reading this strand. One bit of sequence from the HIV genome codes for 4 different proteins.
1 gene, 1 protein.
Yeah, that's the rule. One gene, one gene, one protein, or 1 gene, 1 enzyme, right? Clearly evolved by random, right?
They're still probably going to teach that phrase for another 5 years.
Something like that. I've been teaching it since I was around. I mean, we knew it wasn't true when I was in grad school. Back when I was in grad school. So it's actually kind of interesting. Here's your commercial, here's your commercial break. I think we'll end on this. So when I was in high school, Ronald Reagan was president. I forgot my cane today. Right? You guys weren't even born. Was anyone born when Reagan was in office? Okay, so I'll tell you back then, I can remember very clearly I was in U.S. government class. Were you guys all required to take U.S. government senior year?
Your favorite, actually.
Your favorite class? It was misery for me, I hated it. Accounting was the only worst class. At one point, so the teacher kept trying to avoid what seemed to be this huge, huge discussion going on in the U.S. at the time. The teacher seemed to not want to talk about this new pandemic called AIDS, which was appearing to strike our planet, our Earth. We didn't know where it came from. And at one point, I think I forced the issue in the discussion, and we were doing a public health thing, and I said, look, there's a news report that just came out, and there are 12 people in the United States that have been confirmed to have AIDS. If we don't do something about this now, it's going to become probably the #1 health issue for our generation. It's probably a huge human rights violation, but we ought to take those people, sequester them away until we can figure a solution, and find out who they've had sexual contact with. And the arguments at the time, you guys probably don't know this, but the arguments are at the time, it's pretty offensive, but were, this is a gay disease, because at the time it was not really found in the female population. It thought it was from male to male. And there were certain people in my class who said, you know, this is God's will upon those people, showing them they're wrong. I found it offensive at the time. And, you know, maybe you think it's okay. I'm not going to call you on that, but the U.S. government chose to not do anything about it, and we're now in our current situation on HIV. At the time, the reason I raise this issue is the virus is, if you got infected with HIV, you had a year. And this thing would just ravage your immune system and you were dead. A year and you were gone. And what I saw happening is the virus just jumped into us from monkeys. As it becomes better adapted to us, it's going to kill us faster, and it's going to be able to spread faster. Maybe it's just in the gay population now, but at some point, some gay guy is going to have sex with a female, because there are people who go both ways, right? It's going to get into the normal hetero population, and it is a concern we all have to have. People were not happy to hear that, and people dismissed it in other ways. Some people were concerned about it. It turns out I was wrong, and I can explain why I was wrong. What happened is, people that got infected with the virus accumulated mutations in their body. So the person didn't have just one, here's the sequence of the virus in me. They had 100 different sequences of virus in them. And if they went and had relationship with someone and infected that other person, this person might get a really nasty virus, and in a year they're dead and gone. But I might have gotten mutations in one of my viruses that when I pass that on, the person who gets it, they get infected, and they don't even know for a year that they've been infected, and then they get a little bit sick, and then they realize after a couple of years, they're infected with HIV. So what happened in the HIV was natural selection. The virus that was really nasty and killed people died. The thing it was infecting died off, and then that virus couldn't be spread. But virus, which had accumulated mutations, it was a really crappy virus and didn't make you very sick, that person might be around for 10 years, infecting a whole bunch of people, not knowing that they were sick and infecting people. Which means the crappy virus has propagated. The reason this is relevant for this is, if the virus accumulates a mutation in that nucleotide, 4 different proteins are affected. The virus becomes crappy in 4 different ways. Or better. Right. But what's happened is now, if you get infected with HIV, it can pretty easily be controlled. You know who Magic Johnson is? A former NBA basketball player, Michigan State hero. He got infected with HIV. When he got infected, I'm like, oh, that's too bad. If I'm going to get his autograph, I better get it soon. He's not going to be around. He's been infected for like 20 years. He got infected with a really crappy version of the virus and has been able to control it.
They said that he doesn't even have traces of the virus in his body anymore.
They can't even find traces of it. If they say he's cured, I say bullshit. It's hiding in the cell latent, and they'll find it when he stops taking the medication.
But that's fine, as long as you keep taking medication, they can't find it.
Well, he's rich enough, he can afford it, right?
Exactly. Only problem
What kind of medication is that?
He's on a whole cocktail. Any drug they can come up with to fight it in any different way, he's taking 3 or 4 different drugs.
There's protease inhibitors and integrase inhibitors and all that stuff.
But the point is, if you can find a way to target the genome, or screw up the genome in some way, you could really affect that virus, because it's got these overlapping codes. We'll go back to overlapping codes, and we'll finish talking about microRNAs on Wednesday. Thanks for coming.